Hello, everyone. Good afternoon. Thank you for staying with us and welcome to our closing keynote presentation. I am Kathy Young from GenScript, and I'll be your moderator for this session. CAR-T cells have drastically improved outcomes for blood cancer patients, and great efforts are being made to develop CAR-Ts that are effective against solid tumors. There is also a growing interest in expanding the application of these amazing cellular drugs beyond cancer to the other disease states. Autoimmune conditions such as lupus and diabetes are a new frontier for implementing CAR-T cell-based therapies. Today, we are thrilled to have invited Dr. Geng from Legend Biotech as our closing keynote speaker. Dr. Geng has over 20 years of experience in drug development in the biopharma sector. In 2018, she joined Legend Biotech, a global commercial stage biotech company focused on developing, manufacturing, and commercializing novel cellular therapies. She is currently the VP and head of early stage drug development at Legend, and she is in charge of R&D portfolio development, translational sciences, and also leads non-clinical safety, preclinical, and clinical pharmacology. In her talk today, Dr. Geng will discuss opportunities for CAR-T cells as therapies in autoimmune disorders. And the title of her talk is Autoimmune Diseases, the Arising Opportunities for Cell Therapies. Again, for our audience, please type any questions you have into the Q&A box, and we will address them at the end of the presentation. Now let's welcome Dr. Geng, and we are all ears.
Hi, I'm Geng. I'm going to present autoimmune diseases, the arising opportunities for cell therapies. CAR-T cell therapy has revolutionized the cancer treatment for B-cell leukemia, lymphoma, and multiple myelomas with sustained long-term remission. Since B-cell play central roles in many autoimmune diseases, CAR-T therapy offers similar promises in autoimmune diseases as in oncology. In this presentation, I will share with you the emerging clinical results and discuss the challenges and opportunities of cell therapy in autoimmune diseases. CAR-T cells are engineered T cells. The patient's white blood cells are collected through apheresis. Chimeric antigen receptors are transduced into these T cells through viral vectors. Engineered T cells are then expanded in culture and reinforced to the patient after patient has complete lymphodepletion. In oncology patient, CAR-T cell recognize and bind to target antigen on the surface of tumor cell, become activate, expand, and kill the tumor cells. CAR-T cell has revolutionized the cancer treatment. The first CAR-T cells was approved in 2017. Since then, the total 6 CAR-Ts have been approved. 4 of them are anti-CD19 CAR-T for the treatment of B-cell lymphoma and leukemia, and 2 BCMA CAR-T for multiple myeloma. CAR-T cells have demonstrated remarkable clinical efficacy with sustained long-term remission. For example, in ALL, the overall response is as high as 80%, and the CR rate in CARVYKTI is over 82% upon first approval. Given its remarkable efficacy over other therapeutics, the CAR-T therapies have moved to frontline. It has been over a decade from the initial patient who were treated with CD19-target CAR-T cells. The long-term follow-up data demonstrate prolonged remissions with minimal long-term toxicities and probably cure and for a subset of the patient. It's a success story in CAR-T for the treatment of hematological malignancy translatable for autoimmune diseases. This is a question people have been asking for a long time. So why autoimmune diseases? Autoimmune disease is a type of disease where our overreactive immune system attacked itself and result in cell or tissue or organ damage. The prevalence of autoimmune disease are high. The autoimmune disease affect 80% of people in US with estimated 300 million people. And autoimmune disease a huge medical need. The current therapies include anti-inflammatory and immunosuppressive agents, as well as engineered monoclonal antibody. However, these therapies are inadequate. Many patients eventually will relapse and demand lifetime management. This is a huge market for this unmet medical need. B cell play a central role in many of the autoimmune diseases. And listed here are the number of autoimmune diseases which have the involvement of the B cell in disease development. And B cell could involve in the autoimmunity from several ways. It could act as antigen presenting cell or produce in the inflammatory cytokines and produce autoantibodies. These autoantibodies may attack healthy cells and target healthy tissues. Given the role B cell play in autoimmune disease, B cell has been an attractive target for target therapies such as monoclonal antibody. Monoclonal antibodies such as CD19, the drug is rituximab, has shown to be effective in treating autoimmune diseases, but many patients become refractory and have a poor prognosis. Incomplete B cell creation seems associated with lack of durable responses. For example, in the LUNAR trial of rituximab, only 50% of patients achieve 100% B cell depletion at week 52 and 78. In the same trial, much more patients who had complete peripheral B cell depletion had completed responses at week 52 and 78 compared to those patients who have incomplete peripheral B cell depletion. Compared to antibody, engineered CAR-T cell could lead to deep and prolonged B cell depletion and may offer better treatment options for treating autoimmune diseases. In the past 2 years, more and more clinical data have been reported for CAR-T-treated autoimmune diseases, and results have shown encouraging clinical efficacy in several autoimmune diseases. The first notable clinical data published in 2022 from a German academic study. In this study, 5 SS patients were treated with CD19 CAR-T cells at 1 million per kg. And similar CAR-T expansion kinetics has been observed as in B-cell lymphoma. CAR-T expansion led to deep B-cell depletion. The remission of the SLE was achieved according to Doris criteria in all 5 patients after 3 months. Anti-double-stranded DNA antibody were not detectable after month 3, and with a dramatic decrease of other autoantibodies. All 5 patients have shown B-cell reconstitution and within 4 months. Interestingly, the reconstituted B cell are most naive cells, with plasma blast being undetectable, and the dramatic and decrease of memory B cells. The appearance of naive non-class switch B cells and the clearance of circulating plasmablasts and the downregulation of disease-associated autoantibody seem to suggest that CD19 CAR-T cell treatment may induce a reset autoimmunity in this patient. Even though CAR-T cells could deplete both pathogenic and non-pathogenic B cell, the B cell system is rebooted without the presence of pathogenic B cells. And in another case study published in 2024 in New England Journal of Medicine, 15 autoimmune disease patients were followed for up to 29 months. And among these 15 patients, 8 with SLE, 3 with IAM, and 4 with SSC. All the patients have achieved a significant clinical improvement. The 8 SLE patients have achieved drug-free remission measured by DORS criteria, and the SLE disease activity remained absent for all 8 patients up to 2 years. And the analysis of autoantibody after B cell reconstitution, year after B cell reconstitution, confirmed a sustained absence of SLE specific antibody and support the long-term remission of the SLE patients. It is notable that all the patients could successfully stop their immunosuppressive medication without having relapse or worsening of their disease. And the information suggests even after B cell reconstitution for over 2 years, the patient still can remain disease-free for long-lasting remission. What I have been showing is only two the study, and there's more in the reported clinical data on CD19 CAR-T treated autoimmune diseases. Now, what have we learned from this and clinical results? And the data suggests the complete B cell clearance was achieved quickly, most within 14 days, which is similar as we have observed in B cell lymphoma or in leukemia. B cells were generally reconstituted in 3 to 4 months, and the reconstituted B cell exhibited a naive phenotype and without and not autoreactive. A follow-up in CD19 treatment patients indicated preservation of vaccine antibody but sustained diminution of autoantibody and absence of antibody against double-stranded DNA. By depleting in B cell, CD19 CAR-T cell may induce immune reset. Patients were able to discontinue immunosuppressive medication for up to 2 years. And the CD19 CAR-T cells were generally well tolerated with moderate to low CRS. And the clinical data published so far on CD19 CAR-T cells are indeed very encouraging and promising. From the reported clinical data, we also learned a deep and acute B cell clearance may be desirable in autoimmune disease. Unlike in cancer treatment, long persistent B cell depletion and long-term immunosuppressive may not be favorable in the context of autoimmune patients. Deep and transient depletion of B cell may be sufficient for treating autoimmune patients and achieve durable efficacy. Allogeneic CAR-T cells, which could offer deep and acute B cell depletion without concern on prolonged B cell aplasia, may be a good choice. And this is a case report of allogeneic CAR-T cells for the treatment of autoimmune diseases. And CD19 and alpha-beta T was engineered to knock out and track HLA A and B, CRITA and PD-1 gene to overcome immune rejections. 3 patients were treated, 1 patient with refractory in myopathy, and 2 patients with diffused and cutaneous systemic psoriasis. Significant improvement has been observed in all 3 patients and in the clinical response index score, and with observation of reversal of inflammation and fibrosis. Other than CD19, BCMA, which expressed on both long-lived plasma cells and the plasmacytoid and dendritic cells, is another target for autoimmune diseases. CAR-T Therapeutics has been reported their phase 2A results on mRNA CAR-T cell for the treatment of MG. The 7 patients reported in this study exhibit a clinical improvement in the common MG disease and scale at month 6, and indicating BCMA could be a potential target for treat this type of diseases. There are a lot more case studies in autoimmune diseases, and the positive clinical data has been reported in multiple autoimmune disease. And listed here are the reported clinical data and the published references. The target include CD19, BCMA, and CD19 BCMA dual target. Even though the patient number is limited in this case report, in most of the case report, the number of patients are under 10. But however, this still implies a promising and encouraging potential for cell therapy in treating autoimmune diseases. The number of clinical trials in autoimmune diseases using CAR-T therapy has been growing dramatically in the past few years, especially the past 3 years. There are around 40 clinical trials ongoing in 2024, and 50% of them are allogeneic. And those allogeneic platforms including NK, engineered alpha-beta T cells, and gamma delta T cells. The field is expanding quickly. Jeffrey has conducted a neurologic survey on the prevalence of MG and MS. Surprisingly, it has been reported higher than reported prevalence in both indications. The reported refractory rate is 15%, but in the survey, the rate is doubled. Notably, 50% of those refractory patients are CAR-T treatment eligible, and the survey supports a significant market potential for CAR-T cell. Even though we have observed promising clinical results in multiple autoimmune diseases, the number of patients in this study are very limited. The overall and long-term benefit of cell therapy in autoimmune disease are yet to be demonstrated with more clinical evidence. And safety concern it's always a roadblocker for autoimmune diseases compared to oncology. The safety concerns are most associated with the CAR-T treatment itself. For example, neutropenia, which is associated with the preconditioning of the patient and using lymphodepletion reagent, cytokine release syndrome, neurotoxicity, secondary malignancy, and infections are all the safety risk associated with CAR-T treatment. In summary, CAR-T therapy is an emerging treatment for autoimmune disease with promising clinical outcome. More and more clinical data suggests CAR-T as a potential cure for autoimmune diseases. However, even though the clinical data is still in preliminary with limited patient number, and it is still a promising therapy for treating autoimmune diseases. But the long-term clinical benefit is yet to be demonstrated. Safety concern could be a roadblocker for widely use of cell therapy in autoimmune diseases. Cell therapy, which could provide deep and durable remission, and at the same time minimize the CAR-T cell associated safety risk, would be a final winner in this game. In the end, I would like to thank my colleague Li Junxin and Zhongyun and Guo Weifang, who helped preparing this and this presentation, and also thank you for your attention.
All right, thank you, Dr. Geng, for the very inspiring talk. Yeah, we can see you on the camera. And thank you very much for sharing the latest clinical progress of CAR-T therapy in treating autoimmune disorders, and also deep diving into some of the underlying cellular mechanism. It's very exciting to learn that CAR-T therapy opened a new avenue of treatment for patients with autoimmune diseases. So hopefully the challenges you just mentioned can be addressed and also well controlled in the near future. We will now open the floor for questions, and we do have a couple of questions here. The first one is, could you elaborate a little bit on the advantage of targeting CD19 BCMA in SLE for cell therapies? For example, comparing to the other approaches like antibody-targeted therapy.
I think I have to show that in my presentation, and CAR-T therapy is able to offer deeper and B-cell depletion, and this in the depth of the B-cell depletion from all the evidence and durability of the B-cell depletion, and could offer clinical benefit. And while over other antibodies such as rituximab and or some other anti-CD20 or CD19 antibodies, they will not be able to have that kind of depth of cleaning of B cells, and usually incompleted, and also it takes much longer time to achieve completed B cell depletion, as has been shown in the presentation. So clearly, there is a significant advantage of CAR-T treatment and in autoimmune diseases in terms of CD19 targets, the overall antibody, and targeting CD19 alone.
Okay, thank you so much. The second question is the immunosuppression 2 years severe enough to be life-threatening?
I'm not a physician, and I think in the context of autoimmune diseases, definitely we would like to minimize the durable or the long-term and immunosuppressive and as much as we could. But again, it depends on the patient's own condition and patient management, as well as the complication associated with this immunosuppressive the conditions.
All right, thank you very much. Next question. What was the location of these studies? Would results vary from location?
Yeah, we see, and well, if you go through the references, you can see the case reports are worldwide, and this initial academic study from Europe, and there's industry and phase 1, phase 2 studies. There are academic trials. There's IIT study from China. I think those are solid evidence to support the efficacies and the potentials of cell therapy in autoimmune treatment.
Great, thank you. The next question. Thank you for the presentation. Comparing to TCR-T and CAR-T treatment, who will be having better potential in treating solid tumor?
Solid tumor. Well, in the solid tumor is a different beast, and we have not seen a significant advance. Well, I would say significant clinical evidence or benefit yet from both CAR-T and TCR-T. So I think, from MA perspective, they all have the challenges. Again, we need more actually data to support which one could be actually, and better for the solid tumor treatment. I think the solid tumor, it's definitely another unmet medical need and requiring for more approaches for different modalities.
Makes sense. Thanks. All right, let's move on. We have more questions coming in. So the next is there any effort to look for a new B-cell target?
I'm sorry?
Is there any effort to look for a new B-cell target for treating? I would assume the question is, are we exploring a new B-cell target to treat autoimmune disorders?
If you look at the development and how B cell involved into the development of the diseases, the CD19 could be contributed to cover probably the larger range, and in terms of the B cell the development of the B cell, as well as contributing to the disease the development. And other targets as the CD19, CD20, or BAF will also contribute to the different stage of B cell development, but it was not as wider as CD19. I would see from that perspective, CD19 could be a better choice if we just want to deplete B cell alone. But however, it's not seen. If you look at the current clinical evidence, and people do use BCMA and CD19 B cell, a BCMA dual targeting, and deplete both B cell as well as plasma cells. That could be actually a much better approach, in my personal opinion.
All right, thank you very much. Next question. Do we know if some autoimmune diseases may be better candidates for the treatment than others?
And can you repeat the question? Sorry, I saw like.
Yeah. Do we know if some autoimmune diseases may be better candidates for the treatment than others? In my understanding, probably the efficacy of CAR-T, does it vary between different type of autoimmune diseases? Is there better disease indications that we will see better efficacies using CAR-T therapy?
Well, indeed, and if you look at the published clinical result, definitely we see more and better improvement in SLE compared to MS, even though there's actually a case report on MS with 3 patients treated with CD19 cell therapy. But however, the clinical improvement was not as significant as SLE. I think autoimmune disease are very complex and diverse. It also depends on the stage and of the disease and the subtype of the diseases. I think that's a pretty complicated question. I might not be the best person to address that question, but I think, yeah.
Yes, thank you. Thank you for sharing your insights. The next question. Great talk, thanks. Naive question. Is there a good understanding of the underlying course of autoimmune diseases, say SLE? Would it be expected that autoimmunity would return in some patients even after immune reset? Would you expect autoreactive B cells to become active again?
Well, that's a very good question. This is still the unaddressed question, and we are still looking for the answer with more clinical evidence coming. There indeed, there are two the recent case that the two patients relapsed, one in the Kyverna study and one in another academic study. And the two patients who were treated with CD19 CAR-T cells, they did relapse even after their initial complete responses. And the pathogenic B cell may come back and still produce autoantibodies. So the exactly reason and for those relapsed patients is still unclear, but there might be a chance that if the pathogenic B cell come back, those still autoantibody will be generated. If you look at the data, we did see the complete clearance of anti-double-strand DNA antibodies, but not complete clearance of all the autoantibodies. Some of those autoantibodies are generated by the plasma cells, and probably in the well, B cell can only clear the CD19 positive B cell, including the plasmablast and the short-lived plasma cells in circulation, but they are not able to clear the long-lived plasma cells in bone marrow. So that's why adding BCMA maybe help to completely remove all the autoantibodies. But again, this is a hypothesis and yet need to be demonstrated and supported with more clinical evidence.
All right, thank you very much. The next question, are similar strategies being pursued for autoimmune diseases affecting central nervous system? Are we adopting similar strategies or exploring similar strategies for autoimmune diseases affecting central nervous system?
Well, I think CAR-T is targeting the root cause. I mean, and the attacking of the central and the nervous system is the outcome of the autoreactive antibodies. And whether that course is reversible after the complete removal of the autoantibodies, and that is still not being addressed yet. Maybe our own body have that the strategy to be able to fix certain actually tissue damage in certain degree. But again, I'm not a physician. My personal feeling is that if the severeness of that damage has been in there, probably is not that will not be able to reverse. But however, if we'll be able to eliminate the source and the root cause of the diseases, we may be able to prevent that kind of damage to be further developed.
All right, I think we have two more questions here. So the next would be, what is the biggest hurdle in developing allogeneic CAR-T cells for autoimmune diseases?
Well, in the allogeneic and CAR-T cells, what cell therapy typically associated with HVGD strategy. And in this strategy, and people tend to use anti-host and T cell or an NK cell strategy in order to overcome the host immune rejection. And that is probably not favorable in the context of autoimmune diseases, since it may add additional infection risk. So in allogenic cell therapy development, we're really looking for HVG strategy with some kind of actually, different approaches.
Okay. All right, thank you. Another question is, are there any positive clinical result in treating multiple sclerosis using cell therapies?
I sort of touched on that. There's a case report from Kyverna, and the 3 patients have been treated with CD19 CAR-T cells. But however, the clinical improvement was not as significant as what had been observed in the SLE patients. We are looking for more clinical and data.
Sure, looking forward to that. We have one more question here in the box. I will read it out. I'm not sure if I fully understand, but yeah. Dear man, how can we search similar T-cell engineering from different techniques to other publications which represent CD19 and CD56?
I think I don't think I understand the question either, but I guess that the question is is there any because people using CD59 for allogeneic actually approaches, maybe that's the question you're asking. So is that combination and works? And I think there's some data coming, and I know somebody's working on using that approach in allogenic cell therapy. We are looking forward to their publish the result, if any.
Okay. All right. I think that's all the questions we received so far. And if you have any further questions, you can continue to type and we can get back to you offline. All right, thank you, Dr. Geng, again for the wonderful presentation and answering all the questions. And also thank you to all participants for joining this session. CAR-T and other cell therapies indeed have shown their unlimited potentials in treating diseases beyond cancer. So we really look forward to hearing more progresses of cell therapies in autoimmune disorders as well as in other disease applications. All right, I think this concludes our closing keynote speech. Thank you all again for participating, and please stick around as we will soon begin our award winner announcement. Thank you.
Thank you. Bye-bye.